today we are here to discuss about a new practical you have in your term 1. This time it is, you can see the title, that test for carbohydrate or protein or fat in a given food sample. So this is the title and you have to write it for term 1. Okay, and you have to perform it for the 2 marks that is having in your term 1 examination, that is 4 plus 4 plus 2, 4 for this titration, 4 for the inorganic part, and 2 in the 2 they are having some section, there is the preparation is also there. With that, this the detection of carbohydrate, protein or fat in a given food sample is there. Okay, so it is easier one. For this I have chosen it, but I will give you a sample where you will got that the format you have to write for the preparation, that is the salt preparation, that is for chrome alum and the ferrous more salt, etc. I will give you this PDF. From there you can write it. And today's part, that what you have to write in your copy, this portion, this experiment, I will write on the board and you will copy the same and you will just write it in your lab time. Okay, so what I will be writing on the board, you have to write it, because or take any snapshot or any other method, but you can write it in your copy directly. Yes, this is content based experiment. This is under content based experiment for 2 marks. Okay, so test for carbohydrate, protein and fat. Now I am saying that fat will be neglected, it will be excluded, because the test for fat will be difficult one and also we have to use hard chemicals. But when you will write it, it will be included there. But in exam it will not be given. Only the carbohydrate and protein will be given an alternate. So you have to perform any one out of that. It will be solution A or solution B. Okay, so you have to perform any one out of that and you have to write it in your copy. This is for 2 marks, written it. No, this is not for in channel 5. You have to write it in your main lab file. This you have to write in your main lab file after the inorganic part. After the inorganic portion, you have to write. Now what you have to write, I will tell you and also write on the board. Okay, so first make a column like that: experiment, observation and inference. It is the general column. Now in this section you will know about some new kind of reagent. Okay, this is the question for Harvard. Experiment number 1, that is test for carbohydrate. At first, carbohydrate. Okay, first test is called Mollis test. MOLISCH. This is done by Mollis reagent. What is Mollis reagent? It is the solution or alcoholic solution of alpha naphthol. It is the alcoholic solution of alpha naphthol. So Mollis test, you will be supplied with a liquid sample, that is sample solution, plus you have to add this 1 or 2 ml Mollis reagent plus 1 ml concentrate H2SO4. 1 ml concentrate H2SO4. You can got a ring. That is of red violet color. This ring indicates that red violet ring. This ring indicates that carbohydrate present. There is no probability, it is confirmed. Carbohydrate present. This is test number 1. Okay, next test number 2. This is the failing test you have to perform. 1A, B, failing test. So again have to take the sample solution in a test tube. Sample solution plus failing A plus B or 1 and 2, failing 1 and 2 plus heat in water bath, red PBT, carbohydrate present. B and then C. The test C is Benedict test. Benedict test. Benedict test is also performed by the reagent that is called Benedict reagent. Okay. It is prepared by sodium citrate, sodium carbonate, okay, and then copper sulfate. This is a time taking process, Benedict reagent. So Benedict test, sample solution plus 1 to 2 ml Benedict reagent, write it quickly, 1 to 2 ml of Benedict reagent plus keep in boiling water bath. Keep in boiling water bath. It will give you a reddish PPT, reddish coloration or PPT that confirms carbohydrate is present. These three tests you have to write if your food stuff contains carbohydrate. Okay, when it will be completed, inform me, then I will go for the test of protein. This you have to write in your lab file. Okay, or take any snapshot, it will be faster. Completed Molly's test, failing test, Benedict test. Completed. Now, this is not Molly, it is Molly's. M-O-L-I-S-C-H. Write quickly. Completed. Why are you taking so many time to write just 3 examples, research experiment? Okay. Now I will go for the next experiment, that is the test for protein. Test for protein. Here also we will discuss 3 such tests. 2A, that is biurate test, burate test or biurate test. Biurate test, what is biurate reagent? Biburate reagent is copper sulfate solution. Okay. So again the sample solution plus NaOH solution plus burate reagent. Here in observation section it will be blue coloration that confirms protein present. This is number A. After that number B. Number B is that the next mean hydrogen test. Mean hydrogen test. Mean hydrogen test, it is easier. Sample solution plus mean hydrogen solution plus warm. It is also blue coloration, protein present. Number C is xanthoprotic acid. Xanthoprotic acid test. This is the name of this test. Xanthoprotic acid test. Yes, what is the Benedict test? So Benedict test is the same. Test is very simple. That Benedict test, that take the sample, now add Benedict reagent and warm it in a water bath. That the water bath will be boiling. Place your test tube on this water bath. It will give you that reddish color. This is the Benedict test. Now the preparation of Benedict reagent is difficult. It contains sodium carbonate, it contains copper sulfate, sodium citrate, etc. Okay. Next is that xanthoprotic acid test. This is sample solution plus 1 to 2 ml concentrate nitric acid plus heat, yellow coloration or yellow PPD. This confirms that protein is present. So these three tests confirm you that protein is present in your foodstuff or not. What is the chemistry involved in between this? I will discuss it on another class. Mean hydrogen test. Yes, second one is mean hydrogen test. Complete quickly. Second one is mean hydrogen. Third one is xanthoprotic acid test. Some more test is there, but I have just those. I am selecting those which is easier. Okay. Next is that test for fats. If you have completed, inform me. If you have completed, then inform me. Okay. Now I will go for the test for fats or oils. After that we have to perform, I have to perform the test and you will be the observer. Okay, test for fat or oil. This is number 3. Test for fat or oil. first one under 3, this will be solubility test. Solubility test. So the organic compounds basically soluble in organic solvent. So here the solvent will be taken. It will be water, alcohol and chloroform. Water, alcohol and chloroform. So as chloroform, it is basically used for the to make senseless. So for this, it is very risk to work with this reagent, chloroform. So solubility test basically it is done with alcohol. That sample plus alcohol dissolves. Dissolves, that means fat present. Sample plus chloroform CHCl3 dissolves, fat present. Number B is that acrolein test. Acrolein test. There is also translucent spot test, Bowden test, etc. is there. Hubble test. Acrolein test is simple. Though the smell, the fumes coming out, it is the poisonous, that is of acrolein. So this is acrolein test. B. So sample solution. Sample solution plus few crystals of KHSO4. Sample plus crystals of KHSO4, that is potassium bisulfate, plus heat. What will be the observation? It will be that a pungent odor fume or pungent odor gas, pungent odor gas, that indicates fat is present. So these 3 tests will be sufficient for the test of fat or oil. Okay, now write down another one experiment. Spot test or translucent spot test. Translucent spot test. Translucent spot test. The previous was acrolein test. Next is translucent spot test. This is nothing but that fold a paper, okay, and then make a drop, that place a drop of that sample and then unfold it. Suppose you have kept one drop of the sample on a paper, then fold it, after that unfold it. If you see that there is a spot is observed, that is it not evaporate, a spot is observed, that is called translucent spot test. and when it will be dried enough, the the area of the spot will increase even on heating. So this is called translucent spot test. Okay, so this is the experiment, that is translucent spot test. Spot found, the inference is same. Do not to elaborate the experiment, that how it is performed. Okay, translucent spot test, this is just the experiment. Spot found, fat is present. Clear? So that's all about the 4 experiment under the fat. It is not the 4, it is just 3ABC. This is the C. Clear? Completed this one. Then I will show you the test for aldehyde and also the protein. Now it is the time to observe. Okay, everyone completed. Now observe the experiment, and those experiment total it will be 9 experiment that you have to write in your laboratory notebook. Okay. Okay, students. Now at first the test for carbohydrate. Clear? So here we have a sample. So take the sample, prepare the solution, but you will be supplied with the ready solution. No need to do it. Now add water to it to prepare the solution. So at first we have to go for the first test, that is Mollis reagent. So to prepare Mollis reagent, we will require 2 sample. One is alpha naphthol, though it is 2 naphthol. So at first prepare the solution of 2 naphthol. Okay, already the solution is here. So now I will take the sample solution, very little amount. Observe just 1 or 2 ml. I have. Now add this solution. This is the reagent. Mollis reagent added. Now I have to add here concentrate sulfuric acid. So when you have to take this concentrate acid, you have to take the help of this dropper. You can observe the ring. This ring indicates that your sample contains aldehyde. The ring will be in between the junction of these two, that is in between the sulfuric acid and in between the aqueous solution. Okay, so this is the first experiment with concentrated sulfuric acid and the Mollis ring. Observe it. So this is a confirmative test. Now another one test I will perform for this one, that will be with the failing solution. So take the sample solution and then equal volume of failing 1 and 2, then place it on water bath. Okay, so this test we have to perform now. Take a different test tube, take the sample solution. Now add failing 1 and 2 in equal volume to this solution and then place it in the water bath. Till the solution will be heated up, we will go for the test for protein. So these two tests, Benedict reagent test, will be not required, just write this about. Now we will go for protein. So the protein solution already is here. This is egg albumin I have taken previously. Now first test, this is biuret test. What is the reagents required for biuret test? One is NaOH solution and another one is that copper sulfate solution. So we will prepare these two solution, NaOH and the copper sulfate. But it will be supplied to you. You no need to prepare the solution, it will be ready to you. Okay, one is NaOH, another one is the burette reagent. Okay, now in different test tube, take the sample This is protein sample. Now at first add NaOH solution. Then add this reagent, biurate reagent. Observe the color. Observe the color. This is the blue or the deep blue coloration is there. Violet coloration. Okay, this is the first test to confirm for protein. Next we will perform the next test under protein. Okay, already it is transforming to the red color. You can see that the red color transformation of for that carbohydrate. So two tests we have performed under the carbohydrate section. Now the next test under the protein, that will be the next test, that is ninhydrin test. So ninhydrin solution we have to prepare. Just take a little amount of ninhydrin, but it will be supplied to you. It also gives the blue coloration on heating. Take the sample solution. Add this ninhydrin reagent. Okay. After heat it. So when you will heat it, just use the holder. Okay. Now we got our result. This is the blue coloration after adding ninhydrin solution. Okay. So this is also the confirmative test for that protein. Now the test of fat I have written, but as I told you that it will not be included, so just write this experiment as it is included in your syllabus. So what we have done, that is 2 tests for each, one for carbohydrate and 2 for protein we have performed. That is for carbohydrate, this is first of all the alpha naphthol, taking the alpha naphthol solution, and next one the failing solution, and for protein it is the biuret test and with the neat hydrate. Okay, so that all about the experiment. Okay, now you have to write it in your laboratory file, the laboratory notebook you have. Clear?